Hello. Good evening to the student who have joined. Can you let me know if my voice is audible to you? And also, please let me know if you can see the presentation clearly. In the chat box, please let me know if my voice is audible, and also please let me know if you can see the presentation. Hello. Am I audible? Let me know in the chat box if you can hear my voice properly, and also let me know if you can see the presentation. Hello, am I audible? Please confirm if you can hear my voice and also please let me know if you can see the presentation. We will be starting the session from 6 p.m. Till then, please wait. Okay, so it's 6 p.m. I think I should wait for a minute or two. Let's see if more students join. Okay. Okay, so I hope since there is no, complaint about the audio and the presentation, I hope I'm audible and the presentation is clearly visible. Okay, let me just wait for another minute if more students join, otherwise I will start the session. Okay, so I think I should start with introducing myself and discussing the relevant topics. Okay, so just let me make it a big screen. Yeah. So, very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAM course of Medicinal Chemistry, and today it is our week 4 live session. So, the students who have joined, please let me know if my voice is audible to you, and also please let me know if you can see the presentation. Is the presentation visible and also am I audible? In the chat box, can you please confirm if you can hear my voice properly and also if you can see the video and presentation? Okay. No answer. I hope I'm audible and also I hope the presentation is visible, right? Okay, let me know if there is any issue with the audio and the presentation. Okay, so as I was saying, today it is our week 4 live session. So previously we have already conducted our completed our 3 week the 3 weeks course content. Okay, so in the previous session, what we have discussed our already we have discussed around about the basics of the medicinal chemistry. We have discussed little bit about protein, the structure of proteins, and also we have discussed little bit about receptors and enzymes in our second class, and in our third class, which was in the last Monday, what we have discussed in that class, we majorly discussed regarding the different enzyme inhibitors, the mode of inhibitions, the graphs, and all these things we have discussed in our previous class. So today it is our week 4 live session. So before starting the session, let me clarify a few things. So there will be a problem solving session every week on Monday from 6 to 8 p.m. Okay, these sessions are live on Google Meet platform, and this is the link for the class, and the link will remain same for our entire semester. So if any network disruption happens from my side, do not leave the session. Okay, I will try to rejoin as soon as possible after resolving the issue. And the class is open. So if you have any problem with your internet connection or anything, you can just leave and rejoin at any time. Okay. Since I'm recording the session, there won't be an issue. I will be sharing the recording at the end of the session. Okay. I have muted your microphones and cameras so that I can record the session without any hassle. But the chat box is always open. So if you have any doubt, any query, if you want to say anything, you can let me know through the chat box. Satyam, thank you for confirming. Okay, so if you have any doubt, any query, anything, any issue regarding the voice, the audio, the video, anything, just let me know through the chat box. Okay, the chat box we will also use for conducting our session. Okay, for example, for answering during the MCQ parts. So where you have to find the recording and the presentation, you have to go to the SWAM portal of the medicinal chemistry. Okay, where is the course announcements are there. Here all the weekly announcements are posted, right? So on the left panel, you will be finding a problem solving session January 2026. If you click on that part, you will be redirected to a page which looks like this. Okay, this contain all the YouTube links for the previous sessions. Okay, whatever I have recorded, and also there is a presentation drive link, a Google Drive link, which contains all the presentations which I have shown you last. Okay, so from using that particular link and this particular sheet, you can download the class material. Also, you can check out the previous meetings video, right? So this was all about. I already have discussed these things earlier also. So if you have any doubt or any query, remember the mode of communication is our chat box. So please use the chat box and let me know whatever issues you are having during the class or regarding any materials of the course. Okay. So today it is our week 4 live session, as I have already mentioned that in our first class we have discussed about the details of the medicinal chemistry, a little bit about the proteins. What is the protein? What is the structure of amino acid and different proteins, that we have discussed in our very first class. Followed by our second class where we have discussed about the enzyme, the basics of enzyme kinetics, and we have majorly discussed about the different types of receptors like G protein coupled receptors. We have discussed about the ion channels and and kinase gated receptors and so on, that we have done in our second class. In our third class we have discussed about enzymes, inhibitors, different mode of inhibition, the kinetics and graphs, and also we have discussed about little bit about the central dogma of molecular biology, that is the translation, the synthesis of the protein from the amino acids with the help of the nucleic acids like RNA and DNA. So in our week 4, the course contents that are the receptors. Basically we will today we will be discussing about different receptors that acts as a drug target. Also we will discuss little bit about receptor drug interaction, stereochemistry, chirality and nomenclature. Then we will discuss about the so in the biological system, stereochemistry also plays a very vital role. So it is required that you have a little some basic knowledge of the stereochemistry. Okay, so that the course content of today's week. So what how we will doing? We will be I will be showing you around 20 MCQs question. All questions are related to the week 4 course content. Okay. After showing you a particular question, I will be waiting for 2 to 3 minutes and then I will check the chat box. You have to identify A, B, C or D, whatever which of the following is the correct answer for that particular problem, and let me know here in the chat box. Okay, answering in the class, there is nothing like the marks are not given. Okay, but answering is important, otherwise I won't understand that if you are following the class or not. Okay, but you have to be careful while solving the assignments which are posted here. Okay, the weekly assignment and quizzes, you have to be very careful while answering the quizzes, because all the quizzes contain some marks. Okay, that will be counted for your final examinations or to the what your final certificate. But be attentive to the class and try to answer as many questions as you can. Okay, so let me start with the first question. So the first question of problem number 1, it says that which of the following terms is used to describe a drug that binds to a receptor, fails to activate it, and prevents the endogenous chemical messenger from binding? Your options are number A agonist, number B partial agonist, number C antagonist, and number D inverse agonist. Which of the following term is used to describe a drug that binds to a receptor, fails to activate it, and prevents the endogenous chemical messenger from binding? Let me know A, B, C or D, which of the following you think is the correct answer for problem number 1. I will be waiting for 2 to 3 minutes, then I will check the chat box. After checking your answers, I will show you what is the correct answer and we will discuss that particular topic in detail. Okay. I will try to discuss it in detail so that it will be easier for you to answer whenever this kind of descriptive questions are asked related to the same topic. And I know I have already discussed about this agonist, partial agonist, antagonist, and inverse agonist. What are these? I already have discussed, I think, in our second class. So if you have followed that particular class, you can easily answer this question. Let me wait for 2 minutes, then I will check the chat box. Which of the following terms is used to describe a drug that binds to a receptor, fails to activate it, and prevents the endogenous chemical messenger from binding? Agonist, partial agonist, antagonist, and inverse agonist. Let me know A, B, C, or D, which of the following will be the correct answer. The drug that binds to a receptor fails to activate it and prevents the endogenous chemical messenger from binding. What does it call? Okay, let me now check the chat box. I think I have waited for some time. Okay, so the screen is not visible. Others, can you confirm if the screen is visible or not? Anyone? Is the presentation is visible or not? Hello? Can anybody else confirm if the presentation is visible or not? Okay, I got one yes and one no. So if the presentation is not visible to you, you can just rejoin. I think there is some issue with the network because others are saying that the presentation is visible. Nasim, you can just rejoin. Okay, so there is no answer for first question. I hope the screen is visible, right? It should be visible. Okay, there is no answer. Okay, let me show you the answer and then let us discuss it. Okay, thank you for confirming, Satyam. Let us discuss the answer for first question. So, the correct answer for problem number one will be, it is number C, antagonist. Okay. So what is agonist? What is antagonist? So in case of enzyme, we have the term like the active site, and also we have the term like substrate and all these things, right? Or inhibitors. So in case of receptors, the things are a little bit similar, but the terms are different. Okay. So in the case of the receptors also have the active site. Okay. And the active site can bind few drugs or few ligands which are known as agonist, antagonist, partial agonist or inverse agonist. I won't go to the very much detail because I think I have discussed this particular topic already in our week 2 course week 2 I mean class. So you can just go through the PPT if you have any very much doubt. Okay, if your doubt is not clear, you can let me know in the upcoming classes. Okay, so what does the agonist do? So as you can see, there is a very good figure is there. So if this is a receptor, this is the active site of the receptor. Okay, so agonist is the drug that occupy the receptor and it activate them. Okay, so the role of the receptors is to produce some signal with the help of the ligands like agonist, antagonist and all these things, and produce some chemical endogenous messengers to show some activation. Like, so agonists are the drugs that bind to the receptors and activate them. Antagonists are the drugs that occupy the receptors but but they do not activate them. Antagonists block the receptor and activation by the agonist. Okay, so antagonist, as you can see, this red one, it blocks the receptors but no activation happens for the particular receptor. So this is the agonist and antagonist. So what are the differences between agonist and antagonist? So basically, agonist produces an action or response, but antagonist produces an opposite action or response. Agonist is a muscle whose contraction moves a part of the body directly. Antagonist is a muscle whose action counteracts that of the agonist. Agonist is a substance which initiates the physiological response when combined with a receptor. Antagonist is a substance which interferes with or inhibits the physiological action of the another. Agonist is a drug which immediates the action of the neurotransmitter in the brain, and antagonist is the drug that blocks the neurotransmitters. Agonist is the leading character in the literary work. Antagonist is the main opposing force for the protagonist. This is for only just for giving you an example. Okay, so basically these are the differences between agonist and antagonist. So the main point that you need to remember is the agonist that produces the signal or activates the particular receptor fully, whereas the antagonist, it does not activate the receptor and does not and it inhibits the action of the agonist. Okay, so these are the difference of agonist and antagonist. So is the first question is clear? I hope now the screen is visible. Whoever was having problem, please let me know if now the screen is visible or not. Okay, Okay, so I will move to the next question. If there is no issue with the first question, if you have any doubt or any query, please let me know in the chat box. So problem number 2, it says that which of the following terms best describes the amount of a drug required to achieve a defined biological effect? Your options are number A affinity, number B potency, number C efficacy, and number D intrinsic activity. Which of the following terms best describes the amount of drug required to achieve the defined biological activity? Number 1 affinity, number B potency, number B efficacy, and number D intrinsic activity. Let me know A, B, C, or D. Which of the following is the correct answer for problem number 2? Which of the following terms best describe the amount of drug required to achieve a defined biological effect? Affinity, potency, efficacy, or intrinsic activity? I will wait for another 2 minutes, 1 or 2 minutes, then I will check the chat box. Let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 2? Which of the following terms best describes the amount of drug required to achieve a defined biological effect? Let me check the chat box. Okay, I got few answers. Number A, B, and C. Three different options I have received as answer. Okay, so Satyam is saying number B and another I have received number A. Let me check which of the following is the correct answer, number A or number B, or is there any other correct answer? So the correct answer will be, it is number B. Very good. So let us first understand about the terms. Okay. So affinity describes how strongly the drug binds to the receptor, depends on the molecular complementarity of the drug and receptor. Okay. Efficacy, the maximum biological effect that drug can produce. A compound with high affinity does not necessarily have the high efficacy. For example, of the antagonist. Potency, the amount of drug needed to achieve the defined biological effect. The smaller the dose is required, the more potent is the drug. It is possible to have potent drugs with low efficacy. Potency depends in part of the affinity of the receptor for binding the drug and in part of the efficiency with which the drug receptor interactions are coupled to response. And so affinity of the drug is the affinity of a drug for a receptor is a measure of how strongly the drug binds to the receptor. Efficacy is the measure of the maximum biological effect the drug can produce as a result of the receptor binding. It is important to appreciate the distinction between the affinity and efficacy. A compound with high affinity does not necessarily have high efficacy. For example, an antagonist can bind with high affinity but have no efficacy. The potency of a drug is the amount of drug required to achieve a defined biological effect. Smaller doses required with the more potent drug is there. Okay, the drug can be a drug can be potent but have low efficacy that is also possible. And what is intrinsic activity? An agonist binds to a receptor and produces a biological response based on the concentration of the agonist and the fraction activated of the activated receptors. The intrinsic activity of the drug determines its ability to fully and partially activate the receptors. Drugs may be categorized according to the intrinsic activity resulting in the Emax values. Okay. So the correct answer for problem number 2 was potency, that describes the amount of drug which is required to achieve a defined biological effect. Okay. So is it clear or do I need to repeat anything? If there is any issue, please let me know. I will repeat that particular question, okay? If there is no doubt, I will move toward the next problem. Okay. Let me move toward the problem number 3. Anyways, if you have any problem, please let me know in the chat box. Okay. So problem number 3 says, which one of the following statements about the intracellular receptor is true? Your options are: number A, they consist of 3 protein subunits. Number B, they contain a ligand binding site near the C-terminal end. Number C, they contain a binding region for DNA near the N-terminal end. And number D, they are activated by the hydrophobic molecules which are synthesized within the cell. Which of the following statements regarding the intracellular receptors is true? Again, I will repeat. Number A, they consist of 3 protein subunits. Number B, they contain a ligand binding site near the C-terminal end. Number C, they contain a binding region for DNA near the N-terminal end. And number D, they are activated by the hydrophobic molecules which are synthesized within the cell. Which of the following statements regarding intracellular receptors is true? Again, I will wait for 2 minutes, then I will check the chat box. I want all of you, whoever has joined the class, please try to answer the question, okay? Even if your answer is wrong, it's fine, but please try to answer the particular question, otherwise I won't understand if you are following the class or not. Which of the following statements about the intracellular receptors is true? Let me check the chat box. Let's see if there are any answers in the chat box or not. Okay, still I have received no answer. See, even if your answer is wrong, it's fine, but you can at least try to answer the questions. No? Okay. Let me then show you the correct answer and let us discuss little bit about the intracellular receptors. So the correct answer, the statement which is true regarding the intracellular receptors will be number B. They contain a ligand binding site near the C-terminal end. So let me first show you what is the structure. So this is the particular structure. Okay. So as you can see, there are four main domain. There are three main domains. Okay. First is the transcription activating domain, which is near the N-terminal end. Then we have the DNA binding domain, which is in the middle. And at the C-terminal end, we have this ligand binding domain. Okay. So in case of this ligand binding domain, we have an active site. As you can see, there is a particular place where the ligand can bind. Okay. And when the ligand can so what happens, so when the ligand so this is the inactive receptors. In the presence of ligand, the particular receptor, it gets activated, and this is the co-activated protein which binds to the receptors, and gene expression and then target protein in the particular activity is seen here. So intracellular receptors are also known as the internal or cytoplasmic receptors, are protein structure found in the cytoplasm or the nucleus of the cell. Unlike Cell surface receptor, they respond to ligands that are small and hydrophobic or lipid soluble, allowing them pass directly through the plasma membrane. Key characteristics, what are the key characteristics of the intracellular receptors? So basically key characteristics is the direct gene regulation. Most intracellular receptors function as the transcription factors. Once activated by a ligand, they bind to specific DNA sequences called the hormone response elements, or HRES, to turn the genes on or off. Slow but lasting effect because they work by alternating protein synthesis. Their effects typically take hours to days to develop but persist for a long time. Ligand specificity, their ligand include the steroid hormone like testosterone, estrogen, cortisol, thyroid hormones, and fat soluble vitamins like A and D. Types of intracellular receptors. Type 1, their location while inactive is the cytoplasm. Their mechanism, bound to the chaperone proteins, translocates the nucleus upon ligand binding. Example, glucocorticoid receptors, androgen receptors, and prostaglandin and progesterone receptors. Type 2 intracellular receptors, their location is in nucleus, already in nucleus, often bound to the DNA in the active state with co-repressors. For example, thyroid hormone, retinoid acid receptors. General mechanism of action are these. Number one is diffusion. Small and lipophilic ligand diffuse across the cell membrane. Binding, the ligand binds to the receptor, causing the conformational change, often release inhibitory chaperone proteins like HSP90. Dimerization, two ligand bound receptors often join together to form a dimer. DNA interaction, the complex moves into the nucleus if not already there and binds to the specific hormone response element sites on the DNA, and response, this binding activates and inhibits the transcription of the target genes into mRNA, which can be translated into new proteins to alter the cell function. So this is the general mechanism of action of intracellular receptors. So this is the very important topic, the intracellular receptors. If you are given to write a short note on the intracellular receptors, so you can combine all these things, and also don't forget to draw the particular figure. Okay, so as this you can see the statement. So now let us go back to our statements. So first the statement, it says it consists of three protein subunits. So this is not the case, and then it says that they contain a ligand binding site near the C terminal end. This is true. They contain a binding region for DNA near the N terminal end. It is untrue. And they are activated by hydrophobic molecule which is synthesized within the cell. This is also false. Okay. So the correct answer was number B. Now we will move forward to our next question. So problem number 4 says cortisol is dash of the glucocorticoid receptors. Your options are number A agonist, number B partial agonist, number C antagonist, and number D inverse agonist. You have to fill in the blanks. Okay. Cortisol acts as what toward the glucocorticoid receptors? Is it an agonist? Is it an antagonist? Is it a partial agonist, or is it an inverse agonist? Again, I will wait for 2 minutes, then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 4. Cortisol is dash for the glucocorticoid receptor. Is it agonist, partial agonist, antagonist, or inverse agonist? Let me know what will be the correct answer for problem number 4. Whatever you think is the correct answer, you can let me know in the chat box. Cortisol is dash for the glucocorticoid receptors. I will wait for another minute, then I will check the chat box, okay? Let me check the chat box. Let's see if there is any answer for problem number 4. Okay, no answer. Okay, let me show you the answer and let us discuss it. Okay, so the correct answer for problem number 2, problem number 4, cortisol is agonist for the glucocorticoid receptors. Okay, so this is basically and it is known as an unexpected agonist. Let us discuss. So glucocorticoid steroids such as cortisols are clinically used as a anti-inflammatory agent and act as agonist at the glucocorticoid receptors. Okay, so so this is the structure of cortisol. Okay, this cortisol is acts as a agonist for this glucocorticoid receptors and they are clinically used as the anti-inflammatory agent. They have the correct size, shape, and binding regions to fit into the binding site and produce the required induced fit for the receptor activation. Recently, it has been discovered that cortisol acts as an agonist, yet it lacks one of the important binding group, that is the ketone, and have two extra ring that should make it too big for the binding site. So this is the structure of cortisol. Okay, so as you can see, this ketone, which is very important group for binding to the particular active site of the receptor, is missing in the cortisol. Otherwise, but it has two additional ring. One is the pyrazole ring and one is the normal phenyl ring, which is present here. Okay, a crystal structure for the receptor ligand complex was studied and it was found that different induced fit have occurred from the normal. This had resulted in a new channel being opened up for the binding site that could accommodate the extra rings. Moreover, extra interaction with the rings compensated for the loss of the usually crucial ketone group. Normally, a different induced fit would be expected to result for the antagonist activity, but in this case, receptor is still activated. Okay, so basically, this is the normal agonist for this particular glucocorticoid receptors, right? But a particular induced fit of the mechanism happens whenever this agonist binds to the receptor. It is similar to the induced fit or the particular our enzyme and substrate complex. Okay, but when this compound binds, a different kind of induced fit happens and a different kind of accommodation happens with this particular agonist, and the extra interactions with these nitrogens, these rings, they compensate for the loss of the ketone for the cortisol, and still this particular compounds act as an agonist for this cortisol, for this glucocorticoid receptors. Okay, so this is known as an unexpected agonist because basically when there is a different kind of induced fit from the normal on the natural agonist of the receptor, it is expected that there will be an antagonism, but in case this particular receptor still remains activated, so it is also known as an unexpected agonist. Okay, so the correct answer was number A. Now I will go to the next problem. So problem number 5 says when an agonist binds to a receptor for a long period of time, it can result in the phosphorylation reaction occurring. Which one of the following terms is most relevant description for the immediate effect? Your options are number A tolerance, number B dependence, number C sensitization, and number D desensitization. Again, I will repeat. When an agonist binds to a receptor for a long period of time, it can result in phosphorylation reaction occurring. Which of the following terms is most relevant description for this immediate effect? Your options are number A tolerance, number B dependence, number C sensitization, and number D desensitization. Let me know A, B, C or D which of the following you think is the correct answer for problem number 5. Again, I will wait for 2 minutes, then I will check the chat box. When an agonist binds to a receptor for a long period of time, it can result in phosphorylation reaction occurring. What is the name of this particular effect? Tolerance, dependence, sensitization, and desensitization. Let me know A, B, C, or D which of the following will be the correct answer. When an agonist binds to the receptor for a long period of time, it can result in phosphorylation. What is the term that is used to describe this particular effect? Let me check the chat box if there is any answer for this problem number 5. Okay. I got one answer which is saying number B. Let me check if number B is the correct answer or not. Okay, so let us see what is the correct answer for problem number 5. So the correct answer will be, it is number D. Okay, desensitization is the term which is used for for this particular phenomena. Now let us understand what is this kind of desensitization. So desensitization can occur by a number of mechanisms. Okay, some drugs bind relatively strongly to a receptors and switch it on, then subsequently block the receptors for a certain period of time. Thus, they are acting as the agonist and then antagonist. The mechanism of how this takes place is not clear, but it is believed that prolonged binding to of the agonist to the receptor results in the phosphorylation of the hydroxyl or phenolic OH group in the receptor. This causes the receptor to alter the shape to an inactive conformation despite binding to the binding site being occupied. Okay, in the case of this ion channel, this would mean that the ion channel is closed. In the case of G protein coupled receptor, the binding site for G protein is closed. This altered tertiary structure is then maintained as long as the binding site is occupied by the agonist. When the drug eventually leaves, the receptor dephosphorylated and returns to its original resting shape. So as you can see, this is a receptors for this is a example. So this is a receptor. As you can see, this is an agonist. Okay, so when the agonist, this is the resting state, this is the induced fit. Induced fit means the to accommodate the agonist to the active site, the receptor changes its shape a little bit so that it can have a better binding with the agonist. So this is for example, this is an ion channel. It is now open after the agonist is bind. So this is the active agonist, but for after some time, what happened, the phosphorylation occurs. So as you can see, this P, it indicates the phosphorylation, and now the receptor changes its shape, and then now ion channel is closed. So this is the one kind, this kind of antagonism. Okay, so this is known as the desensitization. Once the agonist leaves, this particular receptor will move back to its normal resting state, and the particular thing continues. Now, so on, even when longer exposure to the drug, the receptors or drug complex may be removed completely from the cell membrane by the process known as the endocytosis. Here, the relevant portion of the membrane is nipped off and then absorbed into the cell and metabolized. Receptor endocytosis may also occur after short exposures of the ligand, but in the situation, the receptor is often recycled back to the cell membrane and resensitization process. Finally, prolonged activation of the receptors may result in the cell reducing its synthesis of the receptor protein. Consequently, it is generally true that the best agonists often bind swiftly to the receptor, pass on their message, and then leave quickly. Antagonists, in contrast, tend to be slow to add and slow to leave. Prolonged exposure of the target receptors to an antagonist may lead to the opposite desensitization of the sensitization. This is where the cell synthesizes more receptors to compensate the receptor that are blocked. This is known to happen when some beta blockers are given for the long period of time. Okay, so this is the phenomena of desensitization. Again, if you are given to write a short note, please include the structure, the figure also okay, for so because of this figure also, because this figure provides a better understanding for this particular phenomena. Okay, so this is all about the desensitization. Now I will go to the next problem. Problem number 6 says, when binding data is analyzed with the scattered plot, x-axis intercept is used to estimate what of which of the following. Number A, rate of dissociation, number B, KD, number C, the binding stoichiometry, and number D, none of the above. When binding data is analyzed in the scattered plot, x-axis intercept is used to estimate which of the following. Rate of dissociation, KD, binding stoichiometry, or none of the above. Again, I will wait for 2 to 3 minutes, then I will check the chat box. Let me know. When the binding data is analyzed in a scattered plot, x-axis intercept is used to estimate which of the following. Rate of dissociation, KD, binding stoichiometry, or none of the above. Which of the following will be the correct answer for problem number 6? I will wait for some time, then I will check the chat box. Okay. When binding data is analyzed in a scattered plot, x-axis intercept is used to estimate which of the following: rate of dissociation, KD, binding stoichiometry, or none of the above. Let me check the chat box. Okay, I got another answer which is saying the number A is the correct answer. Okay, let me check if number A is correct or not. So the correct answer will be, it is number C. Okay, so x-axis intercept of the scattered plot is used to determine the binding stoichiometry. Let us understand now what is a scattered plot and why it is important. So affinity we have already discussed that affinity and now affinity can be measured using the process known as the radioligand binding. Okay, so affinity is the how strongly the particular compound can bind. Okay, so a known antagonist or ligand for the target receptor is labeled with radioactivity and is added to the cells or tissues such that it can bind to the receptor present. Once an equilibrium has reached, the unbound ligands are removed by washing, filtration, or centrifugation. The extent of binding can be measured now by detecting the amount of radioactivity present in the cells, tissues, or the amount of radioactivity that was removed. The equilibrium constant of the bound versus unbound radioligand is defined as the dissociation binding constant, or Kd. Okay, so now this is the radioligand plus receptors. Now it creates a ligand receptor complex, and this dissociation constant Kd is known as the concentration of L multiplied by concentration of R divided by concentration of Lr. Now concentration of L and Lr can be found by measuring the radioactivity of the unbound ligand and the bound ligand, right, respectively, because the ligand is radioactive, labeled with radioactivity, and after correction for any background radiation. However, it is not possible to measure the complete concentration of R. Okay, so we have to carry out some manipulation to remove R from the equation. So now total number of receptors present must be equal to the receptors occupied by the ligand or the ligand receptor complex and those of the unoccupied or free ligands, right? So R total equal to this ligand, the R total or total receptors is equal to the free receptors which are unoccupied plus the receptors which are bound with the ligand. Okay. So R total equal to R plus Lr. This means the number of receptors that is unoccupied is R total receptor total minus Lr. Now substituting into the first equation and rearranging, we will have this scattered equation where both Lr and L are measurable. Okay, so bound ligand divided by free ligand or is equal to Lr divided by L, which is Lr now can be replaced by R total minus Lr divided by Kd. If you just put the concentration of L, okay, now with from this equation, if you put the concentration of L, you will get something which is looks like this. Okay. Now we are still face the problem with Kd and R total cannot be measured directly. However, these terms can be determined by drawing a graph based on the number of experiments where different concentrations of known radioligands are used. Lr and L are measured in each case and the scattered plot is drawn. Okay, which is looks like this. This is the scattered plot. Okay, so now this binding stoichiometry or the bound versus free ligand can be given by the this can be this binding stoichiometry can be given by this thing. Okay, so this gives us a straight line where the point mixes x-axis represent the total number of receptors available and the slope measured by the radioligand's affinity to the receptors and kd to be determined. So this is known as the scatter plot. This looks like this. Now A, this A is the radioactive only. This line A, which is corresponds to A, which is the radio ligand only. X is the radio ligand plus the competitive ligand, and Y is the radio ligand plus non-competitive ligand. Okay, so competitive, non-competitive. This is the similar like we have discussed in our enzyme inhibitors, like competitive inhibitor, non-competitive inhibitor. In case of receptors, the ligands, the competitive and non-competitive are the same thing. Okay, and also there is a displacement or inhibition curve that can be drawn, which gives the amount, the value of IC50 for in case of the this particular antagonist. Okay, so this may look like a little bit, problematic, but once you practice it by yourself, you will see it is a very easy. So just you have to do one thing, that you have to substitute this particular so in case of this LR and L, you have to substitute this L by using this equation. Okay, and the R you have to use this particular value, so you will get this particular equation. This equation is very important, you have to remember this equation. Okay, because this is the equation for your scatter plot. And also try to remember this plot number A, which is the scattered plot. Okay, where the intercept in the x-axis, it gives the amount of the this gives the R total. Okay, and in case of the particular this LR by L, it is given by the intercept of the y-axis. Okay, now I will go to the next problem. Problem number 7 says, which of the following terms is the therapeutic index of the drug for humans? Your options are number A, TD50 by ED50, number B, LD50 by ED99, number C, IC50 by TD50, and number D, EC99 by IC50. Which of the following term is the therapeutic index for the drug for humans? Which of the following terms is for the therapeutic index of a drug for humans? Your options are number A, TD50 by ED50, number B, LD50 by ED99, number C, IC50 by TD50, and number D, EC99 by IC50. Let me know A, B, C, or D, which of the following is the correct therapeutic index for a drug in humans? Again, I will wait for 2 minutes, then I will check the chat box. Let me know. Which of the following terms denotes the therapeutic index of a drug for humans? Which of the term denotes the therapeutic index for a drug in humans? Let me now check the chat box. Okay, so I have received two answers, number B and number C. Let me check which of the following is the correct answer, number B or number C. So the correct answer will be, it is number A. TD50 by ED50 denotes the therapeutic index of the drugs in humans. Okay. Now let us first understand what are the particular terms, okay, and what is the therapeutic index. Okay. So the therapeutic index is the quantitative measure of the drug safety calculated as the ratio of the dose that produces the toxic effect to the dose that produces a desired therapeutic effect. The TD50 denotes the toxic effect, and ED50 is the effective or desired therapeutic effect. Higher the therapeutic index indicates safer is the drug, means wider margin between its therapeutic and harmful doses. Okay. The standard formula for the therapeutic index. So this figure also gives a description of the drug safety and therapeutic index. So this is the percentage of responding. This is from 0 to 100%, and there we have a mark for 50%, and this is the amount of drug per kg which is given. Okay. So this is the curve for the therapeutic effect. This is the curve for the toxic side effect. ED50 is the amount or the concentration of drug which is required to produce the 50% of the therapeutic effect, and TD50 is the concentration of drug that is required to produce the 50% of toxic effect. Okay, and therapeutic index is the ratio between the TD50 and ED50. Okay, so the standard formula for therapeutic index uses the median toxic dose TD50 and the median effective dose ED50. The formula is expected as this expressed as this therapeutic index is equal to TD50 by ED50. ED50 is the median effective dose, the dose at which 50% of population achieves the desired therapeutic effect, and TD50 is the median toxic dose, the dose at which the 50% of the population experiences a toxic effect. The preclinical animal studies, the median lethal dose sometimes is used instead of the TD50. Now, high therapeutic index, it just denotes the high therapeutic. So, how when the high therapeutic index will be achieved? When the TD50 is much greater higher than ED50, means the concentration required to the produce a therapeutic effect is much lesser than the concentration which gives rise to the toxic effect, which indicates that the drug has a wide safety margin. A drug with high therapeutic index considered safer because it has a wide dosage range between the effective and toxic dose. Healthcare providers can prescribe those drugs with less concern for close monitoring, as there is a low risk for toxicity as the effective doses. Example, penicillin has a high therapeutic index. The low therapeutic index or narrow safety margin, a drug with a low therapeutic index has a narrow safety margin, meaning the effective dose is close to the toxic dose. Even a small increase in the dosage can lead to the toxic effect. Patients on this medication require careful dosing and frequent monitoring, often through the blood test, to ensure the drug stays in the wide safe range. Example, digoxin, warfarin, lithium are the well-known narrow therapeutic index drugs. So important considerations are the therapeutic window. A related term for the therapeutic window refers to the range of the drug concentration in the blood that provides a safe and effective treatment. Individual variability. A drug's therapeutic index has an average measurement for a population. Individual patient factors such as age, genetics, and other medical conditions can alter drugs' effect. Complex interaction, drug-drug interaction, or the presence of the other substance like alcohol can change the drug safety profile and affects its therapeutic index. Okay. So the correct answer was number A, and these are all the terms, and other terms are there like IC50. So in case of enzymes and inhibitors, the IC50 values are considered, which denotes actually the concentration for the 50% inhibition. Okay, the concentration of the inhibitor which gives rise to the 50% inhibition for the enzyme's activity is known as IC50. EC99 is the similar to the so is the effective concentration for the 99% of the responding. Okay, so these are the some terms which are similar to each other. Okay, so remember what is TD50, what is ED50, and this is a therapeutic index. The graph is very important, so try to remember the graph. Now I will be moving to the next problem. Problem number 8. It says that skills analysis is performed to determine the dissociation constant Kd for dash. Your options are number A agonist, number B inverse agonist, number C partial agonist, and number D antagonist. Skills analysis is performed to determine the dissociation constant of what? Agonist, inverse agonist, partial agonist, or antagonist. Skill analysis is performed to determine the dissociation constant or KD for which of the following: agonist, inverse agonist, partial agonist, or antagonist. Let me wait for some time and then I will check the chat box. Meanwhile, let me know A, B, C, or D, which of the following you think is correct answer for problem number 8. Skill analysis is performed to determine the dissociation constant or KD of which of the following: agonist, inverse agonist, partial agonist, or antagonist. Let me check the chat box. Let's see if there is any answer for problem number 8. Okay, I have received 3 answers and all are saying that number B is the correct answer. Okay, let me check if number B is the correct answer or not. So the correct answer will be, it is number D. Okay, for antagonist, the skill analysis is performed to determine the dissociation constant or KD. So a skilled analysis is used to determine the dissociation constant or KD for the competitive antagonist. Okay, last time we have discussed related to that is the scattered plot. So skilled analysis is a little bit different for the scattered plot. So here this is E observed by E maximum is plotted against the log M. So this is an agonist is first used for the different concentration to activate the receptor, and an observable effect is measured at each concentration. Okay, and this experiment is then repeated several times with the presence of different concentration of antagonists. So the first, this is the something is very important. So first, the agonist is used for the different concentration. Okay, to activate the receptors and have an observable effect to is measured at each concentration. Then the experiment is repeated with the several times with the presence of different concentration of the antagonist. A competitive comparing the effective ratio, now the effect ratio E effective by E maximum versus log of the agonist concentration, the log of the agonist. So here you can see this is the log of the agonist in the x-axis, and in the y-axis it is the E observed by E maximum, the observed effect and the maximum effect. Now the ratio of the observed and maximum effect, it is plotted and produces a series of sigmoidal curve. As you can see, a series of sigmoidal curves are produced, where A is when the no antagonist is present and B to D is increasing concentration of the antagonist. Okay, produces a series of sigmoidal curve where EC50 of the agonist increases with increasing the antagonist concentration. In other words, greater concentration of the agonist is required to compete with the antagonist. A skill plot is then constructed and compared with the log of the reciprocal of the dose ratio of the log of the concentration, okay, of the antagonist. The dose ratio is agonist concentration required to produce specific level of the effect when no antagonist is present, compared to when the agonist concentration is required to produce the same level of in the presence of antagonist. A line produced from these studies can be extended to the x-axis to find the PA2. This is for example, this is the PA2 which represent the affinity of the competitive agonist. Okay, so this is the skill plot or can be used to determine whether different agonists show similar selectivity toward different type of receptors. A PA2 value of the non-selective antagonist acting on a population of different receptor types is determined in presence of each type of agonist. If PA2 values is similar, the indicative that the agonist shows similar receptor activity. Okay. So this is all about the skill analysis. So in case first there is no antagonist, only agonist is there and this plot is observed, and then when the presence of increasing antagonist, as you can see, the amount of EC50 is increasing, means the amount of the agonist is required to overcome the negative effect which is created by the antagonist. Okay, and then what is the dose ratio is a particular dose ratio on the y-axis of log of the dose ratio versus the log of the antagonist. The concentration is plotted, which gives us the Pa2 value, which is equal to minus log Kd, and from this minus log Kd we can determine the dissociation constant for the particular antagonist. Okay, and so this is the very important, what is the significant of the skill plot, and in which cases we can use. So this part is very important, okay, indicating these things, because because skill plots can be used to determine if the different agonists show the similar activity towards a different kind of receptors, and if the Pa2 values are similar, they indicate that agonists show similar receptors activity. Okay, now I will go to the next problem. So problem number 9 says umbrella effect is an example of, your options are number A agonism, number B antagonism, number C inverse agonism, number D partial agonism. Umbrella effect is an example of which of the following: agonism, antagonism, inverse agonism, or partial agonism? Again, I will wait for 2 minutes. Let me know A, B, C, or D, which of the following is the correct answer. Keep answering, even if your answer is wrong, it does not matter, but try to answer. Umbrella effect is an example of what? Agonism, antagonism, inverse agonism, or partial agonism? Umbrella effect is an example of which phenomena? Agonism, antagonism, inverse agonism, or partial agonism? Let me check the chat box. Okay, so I have received two answers. Both are saying that number C is the correct answer. Okay, let me check if number C is the correct answer, or is there any different answer which is correct. So the correct answer will be, it is number B, antagonism. Okay, so antagonism by umbrella effect, some, okay, let me just use the laser pointer. So antagonism, umbrella effect is an example of antagonism. Okay, antagonism by umbrella effect. Some antagonists are thought to bind to the regions of the receptors which are close to normal binding site. Although they do not bind directly to the binding site, the molecule acts as a shield or umbrella, preventing the normal messenger for accessing the binding site. For example, this is the normal or binding site for a particular receptor, and this is a neighboring binding site, and this is an antagonist. Now the antagonist, it is not binding directly to the normal active site, but it is the binding to the neighboring binding site to the receptor, and after binding it creates an or it creates a shield on the normal binding site, so that now the agonist cannot bind to the active site, and the binding site is blocked. So this is and antagonism. So this is an antagonism, and it looks like the antagonist is creating an umbrella or shield over the normal binding site to prevent the normal agonist for to the binding to the active site. So this is known as the umbrella effect. Okay, so umbrella effect is an example of the antagonism. Okay, so the figure is very important, and the figure clearly shows you how the umbrella effect occurs. Okay, now I will go to the next problem. Problem number 10 says raffoxifene is dash of the estrogen receptor. Your options are number A agonist, number B partial agonist, number C antagonist, and number D inverse agonist. Raffoxifene is dash of the estrogen receptor. You have to fill in the blanks. Raffoxifene is what for the estrogen receptors? Number A agonist, number B partial agonist, number C antagonist, and number D inverse agonist. Which of the following will be the correct answer for problem number 10? Raffoxifene is acts as what to the estrogen receptor? Agonist, partial agonist, antagonist, or inverse agonist? Let me know A, B, C, or D. Which of the following will be the correct answer? Again, I will wait for a minute or two, and then I will check the chat box. Raffoxifene is what kind of agent for the estrogen receptors? Agonist, partial agonist, antagonist, or inverse agonist? Let me check the chat box. Let's see if there is any answer for problem number 10. Okay. Okay, I do not there is no answer. Okay, it's fine. Let me show you what is the correct answer and let us discuss about it. So, raffoxifene is, it is an antagonist for the estrogen receptor. Okay. Okay, this may look a little bit critical, but it is fine. So this is now first look at the structures carefully. This is the structure of the estradiol. Okay, and this is the structure of the raloxifene. So, so the 11 beta estradiol is a steroid hormone that affects the growth and development of other number of tissues. It does so by crossing the cell membrane and interacting with the binding site of the estrogen intercellular receptor. Estradiol uses its alcohol and phenol groups to form hydrogen bond with 3 amino acid in the binding site, while the hydrophobic skeleton of the molecule forms a van der Waal and hydrophobic interaction with other region. So as you can see, this is the skeleton for the estradiol, this hydrophobic region. It interacts with the hydrophobic skeleton of the receptors along with the van der Waal interaction, and this phenolic OH group here and the normal hydroxyl group here interacts with hydrogen bonding with several residues. For example, glutamate 353, arginine 394, and histidine 524. The binding pocket is hydrophobic in nature and quite spacious except for the region where the phenol ring binds. This is a narrow slot and only accept planar aromatic ring. Owing to this constraint, the binding of estradiol's phenolic ring, this determines the orientation of the rest of the molecule. The binding of estradiol induces a conformational change for the receptor, so which sees a helical section known as the H12 folding across the binding site like a lid. Okay, and this is not only seals the estradiol into the binding site but also exposes the hydrophobic region called activating region, activating function AF2 region, which acts as a binding site for the co-activator protein. As dimerization has also taken place, the two of these regions are available and the co-activator binds to both of the complete and the nuclear transcription factor. So this is the binding site for the estradiol. Once estradiol binds, this is the H12. So this dimerization happens with the exposure of the A12 regions, which are the hydrophobic in nature, which looks like this, and with this hydrophobic region the co-activator binds. Okay, and the co-activator binds and with the both come the nuclear transcription factor. Now this binds to a specific region of DNA. This now can bind to the specific region of DNA and switches on the transcription for the gene, resulting in the synthesis of protein. Raloxifene, okay, this will be raloxifene, is an antagonist for the estrogen receptor and used to treatment of hormone-dependent breast cancer. It is a synthetic agent which binds to the binding site without activating the receptor and preventing the estradiol from binding. The molecule, this is the structure of the raloxifene. The molecule has this two phenolic group. This has a one phenolic and two phenolic group which mimic the phenol and anchoric group for the estradiol. The skeleton is also hydrophobic, matches to the hydrophobic character of estradiol. So why? This now question arises, why raloxifene acts as an antagonist but not as an agonist? The answer lies in the side chain. The side chain contains an amino group. This is the amino group which is protonated from to form a hydrogen bonding with aspartate 351, an interaction that does not takes place with the estradiol. As you can see, there is no interaction with aspartate 351 in case of the estradiol, but with raloxifene we have this extra interaction. Okay, then interaction. So if doing so, the side chain prolutes into the from the binding pocket and prevents the helix H12 folding over as a lead. As a result, H12 binding region is not exposed and coactivator cannot bind and transcription factor cannot form. Because of this extra side binding pocket, now this AF2 region cannot exposed and the dimerization cannot happen. Because of the dimerization cannot happen, coactivator cannot bind and the transcription is inhibited. Hence, the side chain is crucial for antagonism, must contain an amino group or correct basicity to ionize and form interaction with aspartate T51, and it must be correct length and flexibility to form an amine in the correct position of binding. Okay, so this is how the estradiol binds in case with the estrogen receptors, how it activates the transcription, and how raloxifene acts as an antagonist. All of these are written in and all the figures are very clear to show you the structure. You do not need to memorize the structure but try to understand why the structures there are changes leads to the antagonism, and what is the similarities and what are the dissimilarities. Okay, now I will go to the next problem. Problem number 11 says, which of the following statement is false regarding the configuration and conformation? Your options are: number A different configuration means a different molecule. Number B, changing the configuration of the molecule means rotating the bond. Number C, conformations of a molecule are readily interconvertible. And number D, none of the above. Which of the following statements is false regarding the configuration and conformation? Your options are: number A different configuration means a different molecule. Number B, changing configuration of a molecule means rotating about the bonds. Number C, conformations of a molecule are readily interconvertible. And number D, none of the above. Which of the following statements are false regarding configuration and conformation? Let me know A, B, C or D. Which of the following statements is false regarding configuration and conformation? Number A different configuration means a different molecule. Number B, changing configuration of a molecule means rotating about the bond. Number C, conformations of a molecule are readily interconvertible. And number D, none of the above. Let me wait for a minute, then I will check the chat box. Now we have moved into our stereochemistry part. Okay, I have already discussed that we will together today we will be mostly discussing about one is the receptors, different kind of receptors, the agents that activates the receptors like agonist, antagonist, and also we will discuss about stereochemistry because it plays a very important role in the medicinal chemistry, which we will which we will see in upcoming slides. Let me check the chat box. I hope to find some answer there for problem number 11. Okay, still no answer. Okay, it's fine. Let me check what is the correct answer for problem number 11 and let us discuss about it. So, the correct answer will be the statement which is false. It is number B, which says changing the configuration of a molecule means rotating about the bond. Now, let us understand what is a different configuration and what is a different conformation. So, so conformation and configuration. So, configuration going from one enantiomer to other requires bond to be broken, and conformations of the same enantiomer can be getting one by another by rotation of the bond. So changing the configuration of a molecule always means that the bonds are broken. A different configuration is a different molecule. Changing the conformation of a molecule means rotating about the bond, not breaking them. Conformation of a molecule are readily interconvertible and are all of the same molecule. Okay, so these are the differences between the what is a configuration and what is a conformation. So remember these things. So through configuration, going from one to another requires bonds to be broken, but from going to one conformation to another means only rotation will occur, not the bond break. Okay, so there are other terms which you need to understand, that are stereoisomers and constitutional isomers. So isomer are the compound that contain same number of atom bonded together in different ways. Now, if the connectivity of the atom in two different isomers is different, they are known as the constitutional isomers. For example, this compound has a R group, a OH and a cyano group, but all the way the atoms are connected are different. Here R and cyano are connected with a single carbon, and here OH, hydroxyl and the cyano are connected towards single carbon. So the connectivities are different, but since all the atoms are same, so these are known as the constitutional isomers. If the connectivity of the atoms of the two isomers are same, they are known as the stereoisomer. For example, enantiomers, they are the stereoisomer. Enantiomers are stereoisomer, and so the EZ double bonds. So these EZ double bonds are these all are stereoisomers. The atoms have the same connectivity, but they are arranged differently. So these are known as the stereoisomers. So these terms are very important: configuration, conformation, stereoisomers, constitutional isomers. Okay, and these are known as the stereoisomers are different type like enantiomer and EZ isomers. So now I will go to the next problem. Problem number 12 says what will be the absolute configuration of the epinephrine and pseudoepinephrine. So these are the two stereo centers. You have to identify the absolute conformation of these two centers. Your options are number A 1s 2s and 1r 2r, number B 1r 2r and 1s 2r, number C 1r 2s and 1s 2s, and number D 1r 2r 1s 2s. So these are the RS nomenclature, which is another important topic for the stereochemistry. Again, I will wait for some time. I will wait for 2 to 3 minutes, then I will check the chat box. Try to identify what is the correct answer. What will be the absolute configuration of epinephrine and 1-epinephrine? For the clarification, this is number 1, this is number 2. Okay, this is number 1 and this is number 2. Now identify. What will be the absolute configuration? Let me check the chat box, then I will show you how to do it if you do not know. Okay, I got one answer saying number C is the correct answer. Okay, let me now check if number C is the correct answer or not, and then let us do it together. So, the correct answer will be very good. It is number C. First, let us understand what is the absolute and relative configuration. So, absolute configuration in stereochemistry is the arrangement of the atoms or groups of the atoms that is described independent of the any other atom or group of the atoms of the molecule. Relative configuration is the stereochemistry is the arrangement of the atoms or the group of atoms that is described relative to the other atoms or the group of atoms. Okay. The atomic arrangement independent of the positions and other substituent in case of the absolute configuration, and for example these are the R and S. Okay, and this depends, and the relative configuration depends on the position of other substituent, for example cis trans or Ez. So now how the RS centers can be assigned, this is given like this the details are given here. Okay, so now for example I will just show you one how to do it. So here as you can see this is the number 1 and this is the carbon number 2, right, that I have already mentioned. So now how to identify if which one is the R and which one is the S. So for remember always, if the rotation is in the clockwise direction, so it is the R configuration. Okay, if priority wise, and if it is in the anti-clockwise, it is known as the S. Okay, so here, so now priority, how to give the priority? So as you can see here, the hydroxyl group, this carbon, now the carbon number 1, so there is another hydrogen. So that hydrogen is the like this. Okay, so the hydrogen is above the plane and the hydroxyl is below the plane, and this is on the plane of the paper. So now this group, the hydroxyl group, will be priority number 1 because carbon is bonded with oxygen. Then it will be have this one because they are here. This carbon is bonded to a carbon which is bonded to a heteroatom nitrogen. So this group is 2, and this group, this phenyl group, will be 3. Okay, and hydrogen is always the number 4. So now if we go from 1 to 3, so we will be having 1, 2 and 3. So as you can see, there is an anti-clockwise rotation. But here is the catch. Since the hydrogen, the priority group number 4 is above the plane, we will have the configuration as R, not S. Because if the priority group, so because this R and S will only be followed if the priority group number 1 is above the plane. For example, here, the priority group number 1 is here, 2 is here, and this is the 3. And now if you rotate it 1, 2, 3, it will have a S configuration because priority group number 1 is on above the plane, but here priority group number 1 is below the plane. So so although 1, 2 and 3 rotation gives the anti-clockwise rotation, the conformation will be R. So now this is R, and this, now if we go to the this carbon, you can see here this priority group number 1 is this one, number 2 is this one, and number 3 is this one. If you go to 1, 2 to 3, you will have anti-clockwise rotation which denotes the S, because here priority group number 1 is on the above the plane, so this will remain S. So two number, so that's how you have to identify the RS nomenclature. Okay, this is very simple. Just practice it 1 or 2 times, then you will identify the correct answer. Okay, if there is any issue regarding this particular nomenclature, how to identify the RNS, please let me know via email. I will discuss it in the upcoming class also. Okay. Now let me go to the next problem. So problem number 13 says which statement regarding the drug thalidomide is true? Your options are number A, R isomer is a sedative, number B, S isomer is a sedative, number C, both R and S isomers can be used as drug, and number D, R isomer is a teratogenic. Which of the following statement regarding the drug thalidomide is true? Your options are number A, R isomer is sedative, number B, S isomer is sedative, number C, both R and S isomers can be used as drug, and number D, R isomer is teratogenic. I will wait for a minute or two, then I will check the chat box. Let me know A, B, C or D, which of the following you think is correct answer for problem number 13. Let me wait for a minute, then I will check. Okay. Which of the following statement regarding the thalidomide drug thalidomide is true? Which of the following statement regarding the drug thalidomide is true? Let me check the chat box. Okay, no answer. It's fine. Let me show you the correct answer and let us discuss about thalidomide. So thalidomide is a classic example how stereochemistry governs the biological activity and why having a particular stereochemistry is very much important for the medicinal chemistry. So the correct answer is number A, R isomer is a sedative. So this is the R thalidomide and this is the S thalidomide. So thalidomide was originally marketed as the safe, non-toxic sedative and anti-emetic drug in 1950s and rapidly became popular to counter the effects of the morning sickness during pregnancy. Unfortunately, it was instrumental in one of the major medical disaster of the modern times when it is produced the teratogenic effects of in the developing fetus and lead to the babies born with the standard limbs and other development deformities and so-called thalidomide babies. As thalidomide was considered to have a safe drug at the time, few suspected that it was the cause of the problem, and by the time it was linked to the deformities, it was withdrawn in 1961. 8,000 to 12,000 babies were affected. Consequently, thalidomide gained a lasting notoriety, was the instrumental in the significant tightening of the regulations surrounding the testing of the drug. Despite its notorious past, it has been continued to interest the thalidomide as it is one of the remarkable properties which indicate wide variety of clinical uses. Early on, it was recognized that the thalidomide has an anti-inflammatory property which is useful treating leprosy. In activity was eventually linked to thalidomide's ability to inhibit the synthesis of the pro-inflammatory endogenous cytokine TNF alpha, which is produced by monocytes. In 1998, the drug was approved first for treatment of leprosy. So why this thing arises? Basically, because this R isomer is a sedative. Okay, it is the reason why this particular activity or the thalidomide was useful for the morning sickness. But there is a S isomer. So first, if when it was given, it was a mixture of R and S isomer. This S isomer, it is the reason why this particular drug shows the teratogenic effect. Okay. So, now thalidomide has a raft of other properties. For example, it is an immunosuppressant and could possibly used for the treatment of autoimmune disease or countering immune response to allow the organ transplant to be accepted by the host. Interest in thalidomide as an anti-cancer started when it was found to inhibit the angiogenesis by unknown mechanism. Test shows that thalidomide did indeed have anti-cancer activity and it entered the phase 3 clinical trial for the treatment of renal cancer and multiple myeloma on the basis. Since then, it has been discovered that anti-cancer properties of thalidomide are more complex than the effect of angiogenesis alone. In some patients, thalidomide can boost the immune system by a variety of mechanisms rather than suppress it, and too many account for its anti-cancer activity. As thalidomide can suppress or boost the immune system depending on individual circumstances, it is known as the immunomodulator. Thalidomide appears to arrest the growth of the cells and promotes the apoptosis directly. So these are the thalidomide analogs. So this is the thalidomide, this is the structure, and these are the analogs like lenalidomide and actimide. These analogs of thalidomide have been synthesized with the aim of removing the teratogenic properties. Lenalidomide and the actimide are the two such examples. Both contain an amine substituted substitutions here, which is found to be crucial producing a safer drug. Revlimid entered to the this Revlimid entered to the clinical trials in 2000, was given orphan drug status in 2001 for the treatment of the then incurable disease of multiple myeloma. In 2003, it enters the clinical trial. It is now approved for the treatment of multiple myeloma. So the correct answer was number A, R isomer of the thalidomide is selective, whereas the S isomer is teratogenic. Okay. Now I will move to the next problem. Problem number 14 says separation of the enantiomers can be done by number A crystallization, number B chiral chromatography, number C LCMS, and number D it is not possible to separate the enantiomers. Which of the following will be the correct answer? Number A crystallization, number B chiral chromatography, number C LCMS, or number D it is not possible to separate the enantiomer. Which of the following is the correct answer? Let me know A, B, C, or D. Which of the following is the correct answer for problem number 14? Separation of enantiomers can be done by which technique? Crystallization, chiral chromatography, LCMS, or it is not possible? Let me check the chat box. Let's see if there is any answer for problem number 14. Okay, one answer I have got which says number B is correct one. Let me check if number B is correct answer or not. So, yes, number B is the correct answer. Chiral chromatography is the technique which is used to separate the enantiomers. So interactions even weaker than the ionic bonds can be used to separate the enantiomer. Chromatographic separation relies on the difference in the affinity between the stationary phase, often the silica, and the mobile phase, which is the solvent traveling through the stationary phase known as the eluent, mediated by, for example, hydrogen bonds and van der Waals interaction. If the stationary phase is made chiral with an enantiomerically pure compound, often the derivative of amino acid, chromatography can use to separate the enantiomer. So this is the normal silica which is used for our normal columns to separate the compounds. Okay, but in case of the chiral compound, chiral silica is necessary, which can be made with the use of a enantiomerically pure derivative, chiral derivative of amino acid. Okay, so now how it can be done? So this is the column which has the chiral stationary phase having the chiral silica. Now the racemic mixture is loaded onto the column which has a RNS both compound. Now the compound is forced to the column through the eluent. Now the R enantiomer reaches to, so now depends on the affinity toward the chirality, depends on the chirality, and it gets affinited with the chiral column. Okay, for example here S isomer has more affinity toward our chiral column, so the R isomer travels fast and collected fast. Okay, S isomer has greater affinity so to the chiral stationary phase, so it travels more slowly and requires more eluent, and it will be separated later. So that's how the enantiomers can be separated using the chiral chromatography. Okay. Now I will go to the next problem. Problem number 15 says it is a mathematical question. Okay. When 28 mg enantiomerically pure mandelic acid is dissolved in 1 semi-cube methanol and the solution is placed in a 10 semi-long polarimeter, and optical rotation of minus 4.35 degrees is observed at 20 degrees centigrade. Calculate the specific rotation. Your options are number A minus 15.54, number B plus 155.4, number C minus 155.4, and number D plus 15.54. When 28 mg of enantiomerically pure mandelic acid is dissolved in 1 cc methanol, the solution is placed into the 10 semi-long polarimeter, and optical rotation of 4.35 degree is observed at 20 degrees centigrade. Now calculate the specific rotation. I will wait for 2 to 3 minutes, then I will check the chat box and I will show you how to solve this kind of mathematical question. When 28 mg enantiomerically pure mandelic acid is dissolved in 1 cc or methanol, the solution is placed in 10 semi-long polarimeter and optical rotation observed is minus 4.35. What is the specific rotation? Try to solve it. Let me check if there is any answer for problem number 15. Okay, I got one answer which is saying number B is the correct. 1. Let me check if number B is the correct answer or not. The correct answer will be, it is number C, minus 155.4. Okay, I think now let me show you how to solve this particular question. First, let us understand what is the specific rotation and how the specific rotation happens. So, the rotation of the plane polarized light is also is known as the optical activity. Observation of the rotation of the plane polarized light is known as the polarimetry, and it is a straightforward way to finding out if a sample is racemic or if it contains more than one enantiomer than other. Polarimeter measurements, polarimetric measurements are carried out in a polarimeter which has a single wavelength monochromatic light source with a plane polarized filter, a sample holder where the cell containing the solution of the substance under the examination can be placed, and a detector with a readout that indicates that how much light is rotated. Rotation to the right is given as a positive value, whereas the rotation of the left as the negative one. So this is the polarimeter instrument. So here is the solution of the substance which is kept in a polarimeter cell. This is a detector. This is a sodium vapor lamp, which is this is 0 degree polarization, and this indicate this is the positive rotation. And okay, the rotation denotes as alpha, and the concentration of solution denotes as a C in gram per cc, and this is the length in the decimeter. Okay. The angle through which the sample of the compounds, usually a solution, rotates the plane polarized light depends on the number of factors. The most important one being the path length, how far the light has to pass through the solution, concentration, temperature, solvent, wavelength. Typically, optical rotations are measured at 20 degrees centigrade in a solvent such as ethanol or chloroform, and the light used in the form of the sodium lamp and the wavelength of 589 nanometer. The observed angle through which the light is rotated is given by the symbol alpha. By dividing this value by the path length in decimeter and the concentration in gram per cc, we get a value of the alpha which is specific to the compound and the and question. This is known as the compound specific rotation, and the okay, so this alpha is given by alpha by c, where this is the known as the quoted as alpha d. D indicates the wavelength of 589 nanometer, the D line of the sodium vapor lamp, and 20 indicates the 20 degrees centigrade, and less are the remaining are the variable. So according to now our question, which is given that the concentration is 28 mg in 1 cc solution and the polarimeter is 10 centimeter long, right? So the path length is 10 centimeter means the path length is 1 decimeter. Okay, now we have to convert the the concentration. 28 mg in 1 cc is same as 0.28 gram per cc. Okay, so now the alpha D is equal to alpha by C. So the alpha is given minus 4.35. Now divided by 0.028 into 1 is minus 155.4. So it is the answer. Okay. So that is how you have to solve this particular questions. You have to only be careful for the units. Okay, the concentration unit must be in gram per cc, and the length of the cell is must be in one in decimeter. Okay, so these are the things which you need to be careful with. So this was the problem number 15. Now let me go to the next problem. Problem number 14 says, which is the most stable form conformation for the cis 1,4 ditertiary butyl cyclohexane? Your options are number A boat, number B chair, number C half chair, and number D twist boat. Which of the following is the most stable conformer for the cis 1,4 ditertiary butyl cyclohexane? Boat, chair, half chair, or twist boat? I will wait for a minute, then I will check the chat box. Okay. Let me know A, B, C, or D, which of the following is the correct answer for problem number 16. Which of the following is the most stable conformer for the cis 1,4 ditertiary butyl cyclohexane? Boat, chair, half chair, or twist boat? After a minute, I will check the chat box. So try to answer the question. A, B, C, or D, which of the following you think is the correct answer? Let me check the chat box. Let's see if there is any answer for problem number 16. Okay, there is no answer. Okay, let me show you, and let me show you the structure of the particular compound. So the most stable conformer for cis 1,2,1,4 ditertiary butyl cyclohexane is the twist boat conformation. So this is the cis 1,4 ditertiary butyl cyclohexane. Now this if it was stayed into a normal chair conformation, you will find that in the chair conformation one tertiary butyl group has to be axial and one to be equatorial. How much you try, one cyclohexane will be axial formation. To avoid this, the compound prefers to pucker into a twist boat shape, where this two large group can be done in a pseudo equatorial position. Okay, to minimize the steric hindrance. So this is the twist boat conformation of this cis 1,4 ditertiary butyl cyclohexane, and it has a lower energy than the chair conformation. Now I will go to the next problem. Problem number 17 says, for molecules having similar chirality centers, the number of optical isomers having even number of chiral center is, your options are number A 2 to the power n minus 1, number B 2 to the power n, number C 2 to the power n minus 1 by 2, and number D 0. For molecules having similar chirality centers, the number of optical isomers having an even number of chiral center is which of the following? 2 to the power n minus 1, 2 to the power n, 2 to the power n minus 1 by 2, or 0. Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following is correct for problem number 17. For molecules having similar chirality center, number of optical isomer having even number of chiral center is which of the following? Let me check the chat box once to see if there is any answer or not. Okay, let me continue. Let's see what is the correct answer for problem number 17. So the correct answer will be number A 2 to the power n minus 1. So prediction for the number of stereoisomers, that is the number of optical isomers and mesoforms. So it depends on the following: the number of chirality centers and whether the chirality centers are similar or dissimilar. Okay. For molecules having dissimilar chirality, the number of optical isomer is 2 to the power n, number of mesoform is 0. For molecule having similarity similar chirality center, these molecule are two type: molecule having even number of chiral carbon and molecule having odd number of chiral carbon. For molecule having even number of chiral center, optical isomer, the number of optical isomer 2 to the power n minus 1, and the number of mesoform is 2 to the power n by 2 minus 1. And for molecule having odd number of chiral center, number of optical isomer is 2 to the power n minus 1 minus 2 to the power n minus 1 by 2, and number of mesoform is 2 to the power n minus 1 by 2. So the question was asked that is for the similar chirality center, number of optical isomer of the even number of chiral center. So for even number of molecule, the number of optical isomer 2 to the power n minus 1, and for the odd number of chiral center, it is 2 to the power n minus 1 minus 2 to the power n minus 1 by 2. Okay. So try to remember this table. I know the numbers they seem to be a little difficult to remember, but try to remember the formulas. Okay. Now I will go to the next problem. Problem number 18. Which of the following statement regarding the decaline system is true? Your options are number A, both cis and trans decaline can be inverted. Number B, only trans decaline can be inverted, not the cis one. Number C, only cis decaline can be inverted. not the trans one. Number D, none of them can be inverted. Which of the following statements regarding decalin system is true? Number A, both cis and trans decalin can be inverted. Number B, only trans decalin can be inverted, not the cis one. And number C, only cis decalin can be inverted, not the trans one. Number D, none of them can be inverted. Which of the following is the correct statement regarding the decalin system? Try to answer it. Read the options carefully. Then let me know A, B, C or D. Which of the following you think is correct answer for problem number 18? Which of the following statement regarding decalin system is true? Let me check the chat box. Okay, the live classes cannot be shift because we have a particular schedule first for that we have already chosen the slots. So right now for me it is not possible to change the particular class from this Monday timetable because but it is after the working time, no, after from the 6 p.m. So it is not possible to the shift the classes because already I have chosen this slot because I am also working. So this particular slot has been chosen according to given the slots available for the NPTEL classes. So if it is possible, you can join the classes whenever you are free. Otherwise, the recordings are given to you. The concept is discussed in different videos. It is possible. No, the each lecture video, whatever I show you the question, the particular question I discuss at the same time. Okay, so you want to know that if it is possible to ask questions which is not in that particular week's content, right? So if you have any doubt, your query, you can let me know in the via email. So particular doubt, I will particularly I can solve in the next class. Okay. And it is not possible to shift the classes because that particular Monday slot has been already given by the NPTEL site to me. And but since I understand the issue, but if it is possible to join the live session, it is fine. Otherwise, you can just go through the recorded classes and ask your problems via email or via the discussion forum. And so that's what I can do for you. Okay, for problem, I think I have received one answer, number B. Let me check if number B is correct or not. So, yeah, the statement which is true is number C. Only cis isomer can be inverted for decalin, not the trans one. Now let us see what is the structure. So this is the cis decalin and this is the trans decalin. Okay. So if the ring inversion can happen, if you invert the ring, so it looks like this. Okay, which is quite possible. So inversion of the 6 decalin is possible, but not to the trans one, because if you want to invert the trans decalin, it is impossible to join to different axial positions like this, okay, into the 6-member ring. So it is possible to lock the conformation of the cyclohexane ring by joining two rings together, which is known as the decalin. Two cyclohexane ring fused with a common CC bond. Two diastereomers are possible depending on whether the hydrogen bond of the ring adjacent is cis or trans. For example, cis decalin, as you can see, this hydrogen and this hydrogen are cis to each other, whereas in the trans decalin, these connecting hydrogens are trans to each other. Okay. When a cyclohexane ring invert, the substituents were equatorial became axial and vice versa. It is fine for cis 6 decalin because one hydrogen is axial and one is equatorial. If you invert that particular 6 decalin, the axial one will be equatorial and equatorial one will be axial. But in case of transdecalin, it is not possible because here both our hydrogens are axial position. If we flip it, and the ring hydrogen, the ring carbon bond, carbon carbon bonds has to be the equatorial one, the axial one, which is not possible. Okay, so ring inversion is only possible in case of the cisdecalin, but not in case of the transdecalin. Now I will go to the next problem. The second last problem, it says which of the following compounds are achiral despite having chiral centers. Your options are number A epimar, number B enantiomer, number C mesocompound, and number D diastereomer. Which type of compounds are achiral despite having chiral center? Epimar, enantiomer, mesocompound, or diastereomers? The compounds which are achiral despite having chiral center, epimar, enantiomer, mesocompound, or diastereomer, which of the following will be the correct answer for problem number 19? Let me know. This is very easy and I know all of you can answer it correctly. So I will wait for only a minute, then I will check the chat box, okay? The compound that is achiral having achiral all despite having chiral center is known as which of the following? Epimar, enantiomer, mesocompound, or diastereomer? Let me check the chat box because I know it is very easy and all of you can answer it correctly. Number B, mesocompound. Number C, mesocompound. Number B. Okay, let me check. Yes, mesocompounds is the correct answer. Mesocompounds are achiral despite they have the chiral compound. So definitions are like this: chiral compound, tetrahedron carbon bonded to four different group. Okay, these are optically active and non-superimposable to the mirror image because they don't have any planar symmetry. For example, this one, a chiral compound, tetrahedral carbon bonded to at least two same group. These are optically inactive. Superimposable mirror images are there. Plane of symmetry is present. For example, this. And mesocompounds, compounds having two or more chiral center. These are also optically inactive. Superimposable mirror images can be formed. Plane of symmetry is present inside the molecule. So characteristic optically inactive even though they have the chiral carbon, they have the chiral carbon. Mesocompound do not rotate the plane polarized rise because the optical rotation of one half molecule is exactly cancelled out by the opposite rotation of other half. This phenomenon is known as the internal compensation. Internal symmetry, they must have a plane of symmetry or a center of inversion. The superimposability of mesocompound is identical to its mirror image. There is does not have any enantiomer. Stereochemistry, if a mesocompound has two chiral center, typically they have opposite configuration. For example, one is R and another is S. Okay. Now this is all about the mesocompound. Now the last question of today's session, the most stable configuration for the butane is, your options are number A anticlinical, number B syn periplanar, number C Gauch, and number D antiperiplanar. Which of the following is the most stable conformation of butane? Anticlinical, syn periplanar, Gauch, or antiperiplanar? Let me know A, B, C, or D. Which of the following is the correct answer for problem number 20? The most stable conformation of butane is which one? Anticlinical, syn periplanar, Gauch, or antiperiplanar? A, B, C, or D. Which of the following will be the correct answer? Let me know in the chat box. So let me check the chat box. We are almost at the end of the session. So I will discuss few things. Okay, I have got one answer, number D, which is saying number D is the correct answer. Let me check if number D is the correct answer or not. Yes, number D is the correct answer. Antiplano, okay, is the stable conformation of butane. So as you can see, these are the different conformation of butane. So this is the syn-pleniplanar, which is the most unstable one, having the highest energy. Then we have a synclinical or Gauch, where this Ame group and Ame group, okay, are is in this conformation. And then we have this this is the 60-degree angle. Then we have this particular anticlinical, okay, so anticlinical is like this, it is a more higher energy. Then we have this antipleniplanar, which has the 180-degree. The methyl are 180 to each other. That is why it is most stable one. Then this is the anticlinical synclinical or Gauch, and this is the synpleniplanar. So this is the rotation, how the rotation happens. Okay. So remember, the most stable one is the antipleniplanar, because here methyl and methyl group both are anti to each other, means both have a angle of 180 degree. That is why it is the most stable one. So this was the last question of today's session. So today mostly we discussed regarding our stereochemistry and some receptors like agonist, antagonist, and how the different topics related to the same thing. So I will be sharing this PowerPoint presentation and also the recording of today's class. Now, whoever the students who cannot join the class because of their work or any other problem, so you can just have the recorded class link. I have already shown you. You have to go to the SWAM portal where the weekly assignments are posted, right? So here on the left panel, you will have this problem solving session recording. If you click on this problem solving session recording, if you click here, you will be redirected to a page which contains all the recordings or the YouTube recordings of the each and every class, and also you will have a particular Google Drive link, and that particular Google Drive contain all the week's PPTs. Okay, it is not possible for me, sorry, to shift the class, because this particular classes are already pre-scheduled by the NPTEL side, and also since I am also doing a PhD, so it is not possible for me to have another class to the another time. It is also comfortable for me, and this is the email ID. If you have any doubt, any query, if you cannot join the class, you have seen the recorded lecture and any query or doubt arises, please let me know via email, or you can also let me know to via the discussion forum of the SWAM portal. Okay, I will try my best to answer your queries. So this was the week 4 course content, and if there is no doubt, I will close the session for today. The presentation and class recording I will be sharing to you soon, so you can go through the particular recording. Okay, and you can check the recorded videos, and also you can download the PPTs and you can read it by yourself. If problem arises, please let me know via email. Okay. So if there is no doubt or query, I will be closing the session. Our next class will be on next Monday from the 6 to 8 p.m. Yeah, you can ask your doubt. Let me know what is your doubt. It is not possible to have the PPT of sir's videos because these are the lecture materials, no. So I don't think this is possible to have the PPTs, and you can go through the book. The book I think I have already mentioned that the basics of the, okay, let me show you the fundamentals of the medicinal chemistry. I think the book name is. So it is not possible, but you can just go through this book. Okay, the name of the book is The Introduction to Medicinal Chemistry by Graham L. Patrick. The book you can follow. Mostly, sir follows this book for the present for teaching this particular course of medicinal chemistry, and this book is very informative. I also follow this particular book because it is very elaborated and very easy to understand. Okay, so if you can go through the particular book. The name is the Introduction of Medicinal Chemistry by Graham L. Patrick. Okay, you take a snapshot and you can download. The PDF is online available. If you cannot find the PDF, you can drop an email. I will share the PDF by via email also. Uh, it is not in my hand. You can just drop an email to the authority if they can provide the slides, because the slides I don't also have the slides. so that I am not sure if they will share it or not. Okay, but you can go through the book. I mean, it is better to go through the book because it is very easy to understand and it has a very elaborated topic. So rather than having the PPTs, I think it is better if you can go through the book, because this book is very good actually. So and the PDF is available, so you can download and you can read. Okay. So thank you all for joining, and if you have any doubt or any query, feel free to reach me through the email. I will close the session for today. Thank you all.